have been organizing it. Please carry on. Fantastic. Please carry on. Is that fine?
Yes. We will start our today's session with I'll just share a little bit about the history of CUBE. So CUBE started in the year 2011 in collaboration with Sophia College and Homiaba Science Center, Homiaba Center for Science Education. And since then, collaborators from all different parts of the country, from different collaborators, from different colleges, from different universities, from school till post-graduation, from KG to PG. That's what we call it. Everyone is welcome here to present their work, to do their work. I would also like to invite Kiran. Kiran is here with us to tell a few words on this. Kiran has been coordinating with CUBE for a very long time. Kiran, are you there? Okay, so Kiran is busy with some work.
So Abhijit, I think it will be nice that if you can give your experience, which you started as a 7th standard student or 8th standard student, that will be great.
Yeah, so I joined CUBE when I was in class 7th, getting inspired by my seniors who were at that time working in CUBE. And since then, since when I joined CUBE, it has been a great experience for how I present myself in terms of communication with others, communication with science communication specifically, with different people. I got a lot of exposure to communicate with different types of people, and that helps a lot when you're going to pursue science as your future, and or in any field if you're going to. So this development can only happen when you are collaborating with others. This collaboration only has helped everyone, including me, to develop so much in terms of speech, in terms of behavior, in terms of attitude, and in every aspect, I guess. Like me, there is Saida as well with us. She has also been there since a long time. Saida, would you like to share something of your experience?
I think CUBE has given me a lot actually. I would like say, in terms of understanding the curriculum better, CUBE has a motto that we always say that we stand for context. There is always context to curriculum. There is collaboration, and so we don't understand alone. And the idea is really beautiful that people from different age group are joining. So everyone has a different level of understanding a particular concept, and when we all meet together, when we discuss, we share um, our understanding, and that development is much better. So instead of learning things in a more textual way, uh, we construct knowledge. We don't take from textbook and be like, okay, this is written, but why it is in such a way that is more impactful. And it has helped me personally in my curriculum a lot. And like Abhijit said, it is not just about curriculum. It is also how we put forward ourselves to others, and So that helps. And as a kid, we have this thing in mind that okay, science is something where you need labs, you need we don't imagine scientists like doing research in a simplistic manner, but that level of understanding we get through Q. So what we build is scientific temperament actually. So yeah, thank you. I would like to say thanks to Arunan sir and the Q team, GN sir and Arunan sir especially to start a program like this and helping not just me but students from different parts of the country and different age groups to help us understand our curriculum better.
Yes, thank you, Saida. So our answer is writing in the chat box textual versus contextual. When we have a context, then we develop. Then we can imagine things. So on that note, we will start with our first group. That is fruit fly group. They have some interesting things to share with us that what they have been doing, and I hand over the mic to the fruit fly group, starting with Smriti. Smriti is a second year BSc student from Elphinstone College, Mumbai, and she is with her collaborators. Uh, we'll just start. Just a minute. The screen is visible?
Yes. Yes, it is.
Okay. So I hand over the mic to Smriti and group. Thank you.
So, hello everyone. I am Srujal from Elphinstone College. I am currently pursuing second year in biotechnology. So, hello everyone. Have you ever seen these flies in your house, in your kitchens near the sink? Yes, this is a fruit fly. So, next slide, please. Yeah, fruit fly belongs to the kingdom Animalia, phylum Orthopoda. They belong to the phylum Orthopoda because their body is divided into head, abdomen, and thorax and abdomen. They belong to the class Insecta, order Diphtheria. Order Diphtheria because they have the two pair of wings, and some species belong. Their family is Drosophila, and their genus is Drosophila, and species is Melanogaster. So most of the flies belongs to the Drosophila melanogaster, but not all the flies. Next slide, please. Next slide, please. Am I audible? Yeah. So why fruit fly is considered as a model system? Fruit fly is easy to breed. They are accessible. They are available everywhere. They have short cycle, and they can culture with the minimum requirements. And the life cycle of the fruit fly gets complete into 8 to 9 days. So we can obtain a large number of flies with a single female fruit fly. So how to trap the fruit fly? So for trapping the fruit fly, we just need the 500 ml bottle and the bed. We can use fruit as a bed for bed. We can use any fruit as a bed. So we have to just keep the fruit in the bottle of 500 and where you see more number of flies, you have to just keep the bottle there. The flies will be automatically attracted. And the another way of trapping the fruit fly is that you can keep a plate. In that plate, you have to just keep the bread. You can use any bread as a fruit, and the flies will be attracted to that. So this is a way how you can trap the fruit flies. Next slide, please. Next slide, please. These are the photos of the trapping what we have used for trapping the fruit flies in our homes. These are our home labs pictures. So, and one interesting thing to know about fruit fly is that fruit fly has won 6 Nobel Prizes. You can't believe the fruit fly has won 6 Nobel Prizes till now. So, the first Nobel Prize was won by the T.H. Morgan in 1900 for the discovery of the role of the chromosomes in heredity. In 1946, H.J. Muller has received the Nobel Prize for the production of the mutation by the means of X-ray radiation. In 1995, Louis Christian has won the Nobel Prize for the discovery of genetic control of early embryonic development. In 2004, RXL, along with the Linda, for ordering receptors and has received the Nobel Prize for the olfactory senses. And recently in 2017, in 2017, Jeffrey C. Hall, Michael Rauschenbach, and Michael W. Eng has received the discovery for the mechanism of molecular mechanism of circadian rhythm. So, what is the circadian rhythm will be explained by Smithy. Over to you, Smithy.
Thank you, Shrujal. Good evening, everyone. So, the latest Nobel Prize was won by the latest Nobel Prize was won by Jeffrey C. Hall, Michael Rauschenbach, and Michael W. Young in 2017 for the discoveries of molecular mechanisms controlling the circadian rhythm. Next slide, please. Now, as we can see in the video, cubist Aswathi from Kerala kept a tomato on a white sheet of paper in August of 2020, observing the number of flies visiting the tomato in the day and night. In the Cube Chatshala screenshot at 45 seconds in the video is a graph based on 5 days of continuous experiment elaborating more about the fly's activity pattern of day and night that can be seen. Only a few number of flies were observed from 10 p.m. to 6 a.m., while maximum number of flies were observed in the afternoon and evening, which is shown as 2 peaks in the graph, which will be seen later. Next slide, please. Although variation in the number of flies is observed during the 5 days of experiment, still a pattern is followed where flies were found to be more active in the day and less in the night. Next slide. Now, while observing the activity pattern of fruit flies, cubist observed pupae in the bait container. So we thought, why not culture them in home labs? And to do so, we require a nutritious media, which Smithy here will explain to us. Over to you.
Yeah, thank you, Vaishnavi. So for culturing fruit fly, we need a media, and in college lab, we use a standard media, that is standard corn meal agar media. And at home, we don't have the components for that. And So in this corn meal agar media, we add maize powder, yeast extract, sucrose, dextrose, propionic acid, and orthophosphoric acid. Also agar to solidify the media. But we don't have all these things at our home, so instead of that, we try to make an alternative of corn meal agar media. And Cubist started using tomato. As you could see, the tomato was kept and the larvae started coming. So tomato was used also because it can provide us with the nutrition that yeast extract does, like the protein and the vitamin stuff. So then we added rava to it. Rava as a solidifying agent instead of agar. As a like we don't get agar at our home. You can't get it. It's kind of expensive. Then sugar. So instead of sucrose and dextrose, we are using sugar at home, the table sugar that we add in tea and coffee, water for binding all these things together, and vinegar to avoid contamination, like as an antifungal or antibacterial agent. But when I joined Cube, I didn't knew how to culture fruit fly or how to make a media, and I was told to trap and culture fruit fly. So I went through some YouTube video and I came across a video of David R, where he explained how to make a media. And he was using a potato, like smashed potato or instant potato. It was in a pack, and banana with some vinegar, apple cider vinegar, and yeast powder. But again, I didn't had all these things. So instead, I used. Abhijit, can you please change the slide? Yeah. So instead of that, I used ripe banana. I didn't had instant potatoes. I used boiled potato and custard apple, which was half rotting just to attract the flies, and 1 teaspoon of vinegar. So you can see here, like the media is there below and the white paper, so that fly can sit on it. And I got around 80 to 100 flies from the dustbin nearby, and we called it BPCV media. But then after 2-3 days, the media started becoming liquidy, like you can see in the pictures. Uh, Change the slide, please. And like I did this on 8th of October. The media was proper, thick, and then it started becoming liquidy. A liquid layer started forming there on 9th. And slowly, as it was becoming liquidy, if the fly sits on it, the wing will get wet and they can't fly. They will die eventually. So we started improving our media. So from BPCV, I made BPSRB, that is banana, potato, sugar, rava, vinegar media. So I added a little rava to it, and Abhijit changed the slide. So yeah, I added a little lava to it to make it solid. But then again, after a few days, it started becoming liquidy, and as the bottle was kept inverted upside down, the media fell down. So we had a discussion on this, and we came up with a new media, BRSV media, banana, rava, sugar, vinegar media. And here the components were measured properly. Like we had a rough idea of how to make it. So I used 1 banana, 5 teaspoon of rava, half a teaspoon of sugar, and 5 ml of vinegar. And to bind all this together, we were using water. So there also we have a measurement, that is 100 ml of water, where you boil it first and then put all this banana, rava, sugar, smash and mix them, cook them, and add vinegar to it at the end, and you'll get the media ready. So I've been using this media for past 6 months and it is working. As you can see in the picture, please change the slide. And like there are 5 bottles and a jar, and the jar is kept upside down, but still that like the media which is there, it is not falling down. So yeah, it was working and it still does. So then I trapped those flies, but I still wondered at how do they get attracted towards the bait that we keep, as Sujal was explaining. And to solve that, like to find the answer to that question, I'll ask Saidadi to explain this thing, the olfactory meter, to explain how does the larva get attracted toward the banana smell.
Thank you, Smriti. So, before I'd like to take some points from Smriti before actually diving what is this olfactometer and stuff like that. We got to know that fruit fly was previously cultured in corn meal agar media, and it's been so there is this CSBZ fly, that is Canton Special Benzer fly, that is considered as standard fly, and it has been cultured in laboratory, that is in this corn meal agar medium, for about 70 to 80 years now. So, and the life cycle of the fruit fly is about 10 to 12 days. So it's more than 2000 generation of this CSBZ flies that has been cultured in laboratories. So, because the food is already available to them, and normally fruit flies, in search of food, they use their sense of smell in order to get towards the food. These CSBZ flies don't really need to use that olfactory sense in order to get towards the food, because the food is already present there 24/7. So, in this 2000 generation, there might be some mutation, and our question is particularly regarding mutation regarding olfaction. So, is there any mutation in their olfactory senses? Is what we are trying to find. So, you can, if we wanted to compare the lab bred flies, the CSBZ flies that has been cultured in laboratories for so many years, and with the flies, the wild type flies, like what Smithy is culturing as of right now, and compare these two. And our hypothesis is that this CSBZ flies will have low sense of smell as compared to the normal flies, the wild type flies. Can we go back to the previous slide, please? So in laboratory, what we used to do is we used to take an agar plate, and beneath that we used to keep a reference paper so that it is easier for us to take count. Divided. So here you see a circular image. So we used to draw a circle on the reference paper and divide it in half. One side we are keeping banana smith, and particularly in laboratory we used to use isomyl acetate, and at the opposite end we used to keep water, and we used to keep larvae, particularly second instar larvae, and see and put a timer of 1 minute and see where the larva is moving. We expect that the larva would move towards isomyl acetate. Now, if we dilute this so much, so we wanted to find out at which dilution will the distribution be like 50% each, in the sense what as of control. So let me explain the control part as well. In control part, what we used to do is on both the side we are keeping water and we are seeing the distribution, and we expect if we keep 10 larvae, we expect that the ratio would be 1:1 or 2:1, because it could be that 6 larvae are going towards one end and 4 larvae are going towards the other, because it's a random movement. It is not that they have to be exactly the same, but around equal. That is our idea. So this was olfactometer that was designed by Cubist. Now, of course, we don't have agar at home, so an home lab alternative to it. Can we go to the next slide, please? Abhijit, can we go to the next slide? Yeah, thank you. So, in order to do this olfactory assay at home, what we came up with is, instead of using agar, we can use rava, or people also came up with different ideas like using China grass, but rava was preferred because rava is very common and is available in every household. At one end, we are keeping banana paste, and the other we are keeping water. And so, before starting the actual experiment, like what we used to do in laboratory, we perform the control part. We actually calibrate the device. We need to know whether there's some problem with the device or not, whether this olfactometer is really proper or not. So we keep water at both ends, and we expect that there would be random movement. There would be, the ratio would be 1:1 or 2:1. So once the calibration is done, we used to go ahead with putting banana juice at one end and water at the other, and find out what is the distribution. But in order to actually do this Of course, we have CSDs at flies because it's been cultured in labs, but our home labs, the flies that we have captured from the wild, we need to make single line culture of it. And how do we make single line culture? Smriti will elaborate on that. Over to you, Smriti.
Yeah. So actually, I started making single line cultures to identify the species of fruit fly. And for making single line culture, we need a gravid female. So, gravid means a pregnant female fruit fly. So we isolate her and trap her and culture her separately in a new bottle, in a media bottle. And there we get offspring of the same fly. So we can say that all the flies are from same species. And then we can go for the species identification. And for identifying the species, we need to look at the wing of the fruit fly. So, please change the slide. And like if you look at the wing of a fruit fly, there are some features. Like here you can see yellow color coastal vein. It has two breaks there. And below that, the subcoastal vein, it is incomplete. And then the blue color thing, it is an anal cell. So if you see these three features on a wing of a fruit fly, then they belong to family Drosophilidae. And one of the fruit fly, that is Drosophila melanogaster, they belong to the same family. But to look at the wing, we need something that can give us a good picture of it. And there comes mobile microscopy. And for that, we need some dead flies, a needle to separate their wings, torch, and yeah, torch, brush, white sheet paper, and this is how we set them up. Like a torch will be there, on that there is white sheet paper, then a glass slab, and on that we keep the wing. And on that, like you can see they are using brush for picking up the wing of the fruit fly or the fly, and then you can take a picture of it. And this is how you get a good picture of the wing, and you can identify the species of the fruit fly. And yeah, you can see. Then recently, when I started culturing them, I made a mistake. Like you have to change the media after every 2 weeks, and I kept that media bottle. Like even after changing and transferring flies, I kept the media bottle for about a month. And some small creatures known as mites, they started coming inside. And here you can see a video of it. Like those small dot kind of thing, those are working towards each other. Those are mites. And because of this, most of my flies were dying. And to check like how do they look exactly and how they are killing, we started observing them inside the microscope, and this is how they look. So this was like the one of the major group of that I made. And yeah, till now this much has been done, and we do have some future plans for this. So the future plans and stuff would be explained by Raj. So over to you, Raj.
Hello, good evening, everyone. So we have this context to curriculum map in front of us, which explains us the concepts we are using to study the fruit flies. As we can see in the map, there are various concepts and various things put it in front of us, like the neurobiology, the concept of evolution, the taxonomy, the biophysics, developmental biology, and biochemistry, and other things. But these things are just kept in front of us, and we will be wondering like, what are the practical implications of these things in our fruit fly study. So if we look at in the map, we have this the taxonomical thing,
in this, how we can use it in a fruit fly study. We can, when we capture a fruit fly, we can study the development of its wing, what are the features of the wing, and we can practically apply the taxonomical classification study in our fruit fly study. And the single line culturing, which will provide us the fruit flies having similar characteristics, which will ease up in our further studies regarding to the fruit flies. The next, the biodevelopmental biology. When we capture a fruit fly, it surely will mate with other fruit flies captured, and it will lay eggs, and we will be able to see the many stages of life a fruit fly goes into. That is from egg to larvae to pupae, and finally becoming an adult. And we will be able to study the life cycle and how much time it lives. Thus applying the developmental biology in our daily life with this small yet very useful model organism. And also biochemistry, like preparing the various medias for the fruit fly and having them cultured on it with various nutrition sources. Thus we can sorry sir, the screen is not with me. So using this, we can apply the biochemistry concepts of nutrition and essential components for development and growth, and also genetics by studying various mutation among fruit flies, which is also in our future plans. Next slide. So yes, future plans. So after working with fruit flies, our fruit fly group has various plans to go on with studying this wonderful model organism. The first future plan is to study circadian rhythm among the fruit flies using better methods and equipments. The second point is to study mutations in fruit flies. Like we have heard of Parkinson fruit fly, which is present in Q. Belfinstone. So we would like to study about the Parkinson fruit flies more and make out conclusions, what it is, what are the causes, and what can be the cure of Parkinson using those fruit flies. And third is to study ulcerative colitis, which is mainly an autoimmune disease using fruit fly. So, like we would like to study what are causes of an autoimmune disease and what can be possible cure for such thing. Next slide. So this is a list of wonderful collaborators which have helped us a lot in preparing this presentation. These collaborators help us a lot in carrying out experiments in a diverse manner. And it is because of these we are this successful and we are able to carry out many experiments. Next slide, please. And this is the map of our fruit fly collaborators. Where are they situated? This shows us how diverse our collaborator community is, and surely they provide us very promising results. With this, I would like to thank you everyone for being with us and hearing our talk out. Thank you very much. And now I would like to ask Smiti to take over the mic.
Can we take up the questions? I see a lot of questions in the chat, so can we take up the questions? Abhijit?
Yes, I will take the questions one by one and we'll go through that. So first question was by Hina ma'am from Q Kanpur. She's asking, what do you mean by diptera?
So diphthera, if you divide the word, you can say that the die word will stand for two and tera is for wings. So it says that the fripta has two wings. That's the reason it comes under order diphthera. Yeah.
Yeah, I think Dhanraj is not sure with how you summarize the circadian rhythm of fruit fly. So can you once again summarize the circadian rhythm of fruit flies that you observed?
Can we go on to that slide, please? It would be easier to connect. So what we did is like Vaishnavi explained, we kept an open bait, that is on a piece of paper we kept tomato, and we started observing the number of flies visiting the bait. And so what the general pattern that we observed was that from sunrise till the point of sunset, there were more number of flies coming to the bait. And after sunset, the number of flies reduced. Like, let me give an example that the maximum number of flies that we used to observe is about 100, 200. And here we see the maximum peak is about 300 flies. And after sunset, the number of flies used to go down so much so that it used to go to like 5, 10. By 10, 12, it used to reduce to 0. So the general pattern that we could get was during daytime the flies are awake, maybe that's why they're coming to bathe, and we're getting more flies, more activity, and not just feeding, by the way. They are mating. There is courtship behavior also seen. So and just roaming around on the beds, so much like other behaviors are also seen on the bed itself. So what we did is we compared this data, like we have this data that okay, flies are active during the day and not active during the night. But what do we do of this data? How do we understand what is happening inside the cell of the fruit flies? So for that, we compared it with the 2017 Nobel Prize winning work of Michael Rosbash and Michael Young, and Jeffrey C. Hall, of course. That what happens at a molecular level by our body is, and even humans, we are using fruit fly as a model, so it is implying just to understand a human body better. So we are awake at a certain time of the day and sleepy during the night. Why is that so? Can be answered through the 2017 Nobel Prize winning work.
Okay. So that also summarizes, you think, that the question of Anshu from St. Xavier's College, Mumbai, she's asking, what is the significance of studying circadian rhythm?
So yes, these three scientists, of course, have discovered the molecular mechanism, no doubt. But when we talk of research, it is research. It may be already established, but we are trying to get to understand more. And it's not that the proteins and the genes that they have discovered, that could be just only those proteins and genes are restricted to circadian rhythm. There could be more to it as well. So I hope I have answered the question that when we can understand through fruit fly, we can understand our human body better as to what happens inside the cell that is making us sleep at a certain time and awake at a certain time. And yesterday, I remember during Chatchala, there was a very great example given by Arun and Sir of jet lag. So we often think because the time was coinciding with the sunrise and the number dropped down by the sunset. So we used to think that maybe sunlight has a major role to play in major role to play in circadian rhythm and sleep wake cycle. But in cases of jet lag, when a person say from another country is traveling back to India, they may not follow the time zone of India right away. They'll take some time. They'll have jet lag for a period of time, for a day or two, to adapt to the region they have shifted to. So there is much more to discover always, and by using fruit fly as a model, we can understand how our body functions. So I hope the question is answered.
Yeah. Thank you, Saida. So we'll take
We
one
can
last.
also add, Saida, that circadian rhythm is very important for our health.
Yes, certainly. Um, we have a lot of sleep disorders as well, which can be treated, I believe, if we understand the mechanism better. Then only we can come up with drugs that regulate, that can help in regulating sleep, where the patient is deprived of sleep. I'm just giving an example, but there's much more to it, like Batul ma'am added.
Yes, we'll take one last question by Batul ma'am itself. So she's asking, how can we study ulcerative colitis in Drosophila? What was the basis of that?
Yes, so what ulcerative colitis is? It is an autoimmune disease which causes damage to the wall of intestine, and it causes ulcers and mixed bleed. So we have some studies regarding Drosophila which help in regeneration and the curing of the damaged tissue, which helps healing the ulcers. And as we know that intestine is functioning mainly for absorbing the nutrients one have and one takes through the food, and with the damaged intestinal walls we may not be able to take up the nutrition that better. So with the help of this regeneration of tissue and healing of the intestine, one may be able to overcome the serious effect of these ulcers which are created through this ulcerative colitis. So I guess I'm that. Any more questions, ma'am?
So I see in the chat there are of course a lot many questions, but I guess due to time restrictions we'll move ahead. And our next presentation is on snails, and like we actually saw in fruit flies, I was actually talking about olfactory assay, and there in fruit fly we were trying to understand mutation and see whether the so-called standard fly is actually standard or not. Here in snail, using this olfactory assay, cubists are trying to understand learning and memory. So I would like to call the snails group in order to understand what is this learning and memory and how do we go about understanding it. Over to the snails group.
Hello, Mansi, are you there?
Yeah, I'm here. I can't see the presentation actually. Have you started sharing?
Yeah, it's just for Mansi or for everyone because I've started.
I can't see the presentation.
I can see it actually, sir.
Yeah, it's
Okay.
Okay, so I'll start. Yeah, good evening, everyone.
So I take this as an honor to be presenting my research work, our research work in front of this CUBE National Meet today. So yeah, actually the problem is I can't see the presentation. Hello.
Mansi, can you hear me?
Yeah, I can hear you, but
Do you want
the
to try
presentation-
rejoining? Probably that's the problem, like on your end, because we can all see it.
Yeah, I'll try to do that.
Okay, so I suppose there is some network problem with Mansi, so I shall continue. Hello everyone, Abhijit Singh here from K. Somaiya College of Science and Commerce. Um, so snails, as Saida mentioned, that cubists are trying to understand that how learning and memory takes place in an organism. So we can't just take any other organism and any complex organism to study that, starting from fundamental level. Like first, we need to understand how simple organisms do that, and maybe for that, snail could be a best example for doing so. Uh, the the number of neurons in snails is approximately between 20,000 and that makes it one of the simple organisms to study learning and memory. Uh, so that's why snail we have chosen as a model organism so that we can study learning and memory. And also one of other aspect of this is habituation that connects us with the Eric Kandel's experiment in in the year 2000, for which he won the Nobel Prize. So snails, we study snails as a part of our curriculum in taxonomy class. They belong to the, of course, kingdom Animalia. They belong to the phyla Mollusca and class Gastropoda. And the snails that we are using for our experiments and to understand the research questions that we have to find out the answers to that, we are using the giant African land snails or Lysacatena fulica for that purpose. So with that, we come to the question like how do we culture snails in environment, because they live in a free environment. They live where there is moisture, where there is leafy vegetables. They eat on that. So if we want to do any experiment on them, we need to culture them in our home labs or in our college labs or wherever we want to work with them. So let's hear it from Gaurav from K. Somaiya College of Science and Commerce, that how do we culture snails in our home labs and college labs. Over to you, Gaurav.
Thank you, Abhijit. Okay, so snails are actually very easy to culture and don't require much maintenance. You can easily obtain them in the rainy season, like usually on the roadside, or we can easily find them in places like parks and gardens where there's plants and moist soil. So you can obtain them from there. They have an average lifespan of about 5 to 10 years, and they can be kept in your lab or in your home lab in a cage which needs proper ventilation because they require fresh air. You can, as for the feed, you can give them cucumber, coriander, or cabbage, or any vegetables really. Then but do remember to change the feed regularly, because if the vegetables get rotten or something, then it can attract maggots. then you also have to provide water, or at least a moist environment. You can keep like water in the cage or something, because if the snails dry up, they can go into hibernation. Next slide, please, Abhijit. So snails are hermaphrodites and they lay eggs. You can culture the eggs easily if you just keep the eggs in a moist soil or something and leave them be. They will hatch in about 3 to 4 weeks. And then you can even study on young snails, younger snails, which younger snails, and then that was about culturing snails. Now Isha will talk about the research questions.
So today I'm going to talk about how to induce a change and sensitivity in behavior of snails. So among the most relevant mechanisms of adaptation to heat stress is dormancy during the summer months. The behavioral adjustment has the consequence that snails not only restrict their activity to favorable daytime periods, but to the tolerable seasons of the year. When a snail comes in contact with something edible, it will stop and begin eating. The natural food of snails is dependent upon the species. And also, if the snail encounters something offensive, it will directly respond by retreating to its tentacles or also retreating. Sometimes a snail even will retract its whole body into its shell. A normal retraction sequence is tentacle invagination, lifting of the anterior part of the foot along the midline, and it is continued to the posterior invagination and withdrawal into the shell. You can use these reactions to test a snail sensitivity or check the behavior of the snails. This will be explained in more detail by Mansi. So over to you, Mansi.
Yes. So next slide, please. Okay, so now I'll be talking about how do we actually study and how do we design the experiments in which we'll study learning and memory in Lisa Katerina Fulica. So first of all, as I missed the introduction, I would like to say it again that learning means acquiring new information and memory means retaining that information. So actually studying this in complex organisms is very difficult. So we follow a reductionist approach in which we study very simple behavioral patterns and changes in behavior, and that we do it in snails. Okay, so what we do is actually we have designed some assays, olfactory assays. So first of all, the basic idea is to prove that we have induced a change in the behavior of snails. That means the snails have learned and memorized. So first of all, we design a setup which is called as an olfactometer and an olfactory assay. So first of all, we have to check its reliability, and that is why we perform an olfactory assay for the calibration of the olfactometer. And the second step in this experiment is that to prove that the snails eat coriander, that means they are attracted to the smell of coriander naturally. Okay, so after this, what we do is quinine conditioning. So in quinine conditioning, what is done is quinine, the snails are fed with coriander plus quinine, which induces an aversive behavior in the snails, which I'll explain it to you in detail in the next slides. And the third step in this is then performing the same olfactory assay again and to check the snail's behavior. So what is an olfactory assay actually? So olfaction, now by the word olfaction, you can understand that olfaction means something related to the ability of smell. Okay, so here in this olfactory assay, we are testing the sensing, the smelling ability of of snail. Okay, so we are testing the smelling ability of snail in this olfactory assay. So In an olfactory assay, what we do is we take a plain paper, we draw a circle of radius 10 centimeter with pencil. Okay, and we mark now north and south directions on the assay paper, as you can see in the diagram. And then we mark C1 and C2 points on the east and west side. And on the assay paper, we write the details of the experiments, like the date and the type of assay on the top left corner of the paper. Then a glass plate is placed upon the paper on which a drop of distilled water is placed on C1 and C2 sides both, and then a snail is placed in the center of the circle, and this setup is covered with an olfactometer. Now the olfactometer you can see in the second image. Olfactometer is a simple glass box which is covered with black paper on the lateral sides to avoid the interference of light, and it is open from one side, which is the lower end in this image, and the upper side is kept transparent to see what is going on this in the setup. Okay, so this is the olfactory setup, and then we leave the snail and check its motion, that in which direction it will go. So we also measure the time in which is required for each run of the snail. In every assay, we perform some 20 runs. Okay, so this is the basic idea of an olfactory assay, and what happens is in some time, like 1 or 2 minutes, the snail will be moving in some direction. So when the snail body touches the circumference of the circle, we mark that point. Okay, we mark that point with pencil and mention the number of run. So the assay paper at the end of 20 runs looks like this. Okay, so in the first assay, what we do is we place distilled water drops on C1 and C2 side. So this is for the calibration. So what we expect in this, we want to check that if there is any foreign substance or foreign smell inside the setup or coming from any part of the setup to which the snail is getting attracted. We don't want that. Okay, so that is why to check that we do this control assay, and in this we expect that the snail should move randomly and equally distributed runs should be obtained at the end of the assay to say that our olfactometer is calibrated. So the next step, as I said, is we have to prove that snails are attracted to the smell of coriander. So in this assay, we expect that if the snails are getting attracted towards coriander, then they should move towards the smell of coriander, right? So in this assay, we get majority of the runs going towards coriander side. So for example, out of 20 runs, 16 runs, the snail will be moving towards coriander and 4 runs can be going towards water. Okay, so in the third step, that is after quinine conditioning. So now what is quinine conditioning? Actually, quinine is a drug which is used in the treatment of malaria, which can be obtained in pharmacies and which comes in the form of tablets. So these quinine tablets are crushed and this powder is sprinkled over coriander and given to the snails for eating. Now, this is called as quinine conditioning, and quinine conditioning can be done for 24 hours or 48 hours. Now, in our experiment, we have done both 24 hours and 48 hours conditioning to the snails. So, in 24 hours, the quinine laced coriander is kept for 24 hours in the snail setup, and the same goes for 48 hours. So, after conditioning, what we see is that in the assay, the snail goes mostly towards water side. So it moves away from coriander. So this is an aversive behavior or the change in the snail behavior that we get. That in the first assay, we see that the snail used to go towards coriander for most of the runs. But now exactly the opposite is happening. Now the snails aren't going towards coriander. They are moving away from coriander. So they are going towards water. So this is the data which we have got through multiple assays. And if we represent that graphically, so this is the statistic representation of our data. So the first data in that we can see that the snails have been conditioned for 24 hours, and after that, okay, so in the second step we get that the olfactory assay used to be 0.5 because okay, so olfactory response index, that is ORI. So I'll give some idea about ORI. So olfactory response index is a way to represent the data in a number, okay, so that it can be easily understood. The formula for ORI is C1 minus C2 upon C1 plus C2. So that is basically calculated by the number of runs the snail goes towards C1 and C2. Okay, so in the first assay, we see that the olfactory assay is towards 0.5, and then after conditioning, we get the ORI as 0. So this is a very drastic change in the ORIs too. So, and in the second assay, which has the conditioned snails of 48 hours, we see the same trend, and in this, the behavior is even more aversive. So here we are getting the ORI as more than 0.6, and then after conditioning, the ORI goes down to negative, that is minus 0.4. So this is a change in behavior that we are getting in these assays. So by this, we can conclude that the snails are learning and they are memorizing that there is some change. Okay, so next slide, please. Okay, now Gaurav will explain about the goof-ups that we had.
So, goof-ups are important in any learning process. We learn the most from our mistakes. So, we had also done the olfactory assay in the Somaya College lab, but there were some faults in our experimental setup. So, the first of all, we hadn't constructed a proper olfactometer. So we should have placed a glass tank with sides covered with black paper to block any light coming from outside. We just placed a cardboard box which didn't do that, and there was also a window nearby which through which the light came into the box, and most of the times the snail went to in the direction where the light was coming from instead of the stimulus placed in the box. We also didn't have a large data set because we were only doing this with one snail. We should have gone for more snails. So this tells us how important experimental design is. We should design our experiments in such a way that it would give us optimal results. So yeah, this was about the goofers we had. Now Harushi will talk about habituation.
Abhijit, play that video in like a minute. Hi, I will be talking to you about what we did in our experiment with habituating snails. So, firstly, what is habituation? Let me start by telling you that what a stimulus is. So, anything in the environment that you face as a condition, it could be tactile or temperature, is a stimulus. So, if you smell something, that's a sort of stimulus for your olfactory receptors. So, what we did with the snails and what we did to habituate them is that we used a tactile stimulus in which we tried and touched the tentacles of a snail with a grass blade, and we made sure that we touch it with the same intensity and probably at the same ballpark of the spot. Abhijit, just play that video real quick. And this is like a video from the experiment that I did. What I'll do is just keep touching the tentacle of the snail, and what it does is it tries to contract it because it's not very familiar with the touch, and it doesn't know whether or not the touch is particularly harmful or if it does not matter. So what we observed over time is, as and when you keep doing the experiment, the snail gets habituated, which means it learns that the grass blade touching its tentacle is not the most dangerous touch for it. So the first initial few times it will contract its tentacle, and probably by the 39th, 40th time, it will stop contracting and learn that it is not in fact a harmful touch. And so it can proceed to do whatever else work it was doing without having to worry about contracting its tentacle. Now, how the study was inspired is by an experiment by Eric Kandel, who did a similar experiment in sea slugs. And the only difference was what he did is, number one, his stimulus was different, and number two, he was able to. So sea slugs have a structure which sort of has a siphon. So what Kandel did is he irritated the siphon of a sea slug, which caused the gill of the sea slug to contract, and this caused a sort of, I mean, he wondered why that was happening, why the sea slug would react to the first initial few stimuli and then stop after a while. And then he learned that this was because of habituation, and that the sea slug had learned that the stimulus was not in fact harmful to it. Mansi, in the next slide, will be able to tell us how the graph of the experiment looks like.
Next slide, please. Okay, so if we perform, okay, so this is the data or the graphical representation of the habituation experiments performed by me on 2 different days. Okay, so in this, the right tentacle of the snail was touched with a hair strand. So in the first experiment, we see that the right tentacle stopped contracting. Okay, so first I would like to say that on the x-axis, the number of touches have been mentioned, and on the y-axis, the retraction of right tentacle has been shown. So in the first experiment, we can see that the retraction stopped after 77 touches, and in the second experiment we say that the retraction stopped after 66 touches. And then both the experiments show a similar trend of contraction, of reduction in contraction. So initially there was half contraction, that is 0.5 retraction of the tentacle, and then the retraction slowly and gradually decreases to 1/4, and then after some more touches, then the retraction stops fully. Like the same trend is seen in the second experiment. First half retraction is seen, and then 1/4 retraction is seen, and then the tentacle stops retracting. So this is how we interpret the data. And next slide. So the neuronal basis of this will be explained by Ashrita.
Yeah, hello everyone, I'm Ashrita. So basically till now we've been hearing that the snail basically contracts its tentacles, retracts its tentacles when touched with a hair strand or a glass blade, anything that is a tactile stimulus. So how does this basically happen? We all know that the meaning of contraction of the tentacle basically means the muscles are getting contracted. So how does the muscle basically know when to contract? So this basically works on a neuronal transmission mechanism. So once we touch the tip of the tentacle in a snail, certain chemicals under the skin are activated, and these chemicals now get binded to the receptors which are present on the dendrite present below near the skin. So the dendrite is basically the tip of the neuron. So all this happens via an electrical signal transmission. So why is it an electrical signal? It is because the basis of the transmission is the difference in concentration gradient across the cell membrane, that is the neuronal cell, of sodium and potassium ions. So basically the sodium and potassium ions are exchanged, and because there is an ion exchange, we basically call it as an electrical signal. So once the electrical signal is initiated in the snail, it passes through the cell body, which is the topmost part, then through the axon, which is the middle part, and reaches the axon terminal, the end part, where we have certain neurotransmitters that basically help in transmitting the signal to the next neuron. So why do we have these neurotransmitters over there? So from one neuron to the next neuron, we basically have a minute gap, and because of this minute gap, will not be able to communicate via the electrical signal. So the neurotransmitters